Hello everyone. It's nice to have you today for our first webinar on biochemistry and biotechnology in University of UNIgreen. Welcome, it's really a pleasure to have you online today. I will just share my presentation now. So just a few forward before we go into the seminars. My name is Patrick Gonzalez. I am associate professor at SupBiotech in Paris, and I'm co-coordinator of research and innovation for the UNIgreen University. And my research is on biocontrol and microbiome of soil. If you want to contact me for any question or any information, just you have here my email. So it's a great pleasure and honor to be your host and moderator today. And as a disclaimer, I just want to mention that this event will be recorded and uploaded on the UNIgreen YouTube channel. Feel free to ask your feel free to ask your question anytime on the chat. You will be also able to ask live your question, and if you need a certificate for this webinar, for the attendance to this webinar, just send us a mail to this address here. I would like to give you just a few words about UNIgreen because maybe some of you do not know what is UNIgreen. So UNIgreen is an alliance between 8 higher education institutes across Europe, and it's supported by Erasmus Plus program. The specificity of UNIgreen is that it's dedicated to education and research in the field of sustainable agriculture, green technologies, and environmental and life science. Our goals are to deliver a higher standard of education, foster ambitious research project and collaboration, and provide opportunity for students and also researcher and staff. So what it means for you, it's that there is plenty of opportunity within UNIgreen. If you are a student, you will have access to super or cutting edge courses, so get into this. You will have also access to lab for internships. And if you are a researcher, you will have opportunities to make collaboration, to contribute to proposal, such as Horizon proposal, and to get access to different type of technologies and do also exchanges. So many opportunities. If you are really interested, just contact us. We will be happy to get you into UNIgreen. Concerning research within UNIgreen, so we have divided our interest into 8 axes of research that we are calling GRC for Joint Research Center. So here you have the different axes on which we are working within UNIgreen. And if you are interested by one or several of these axes, again, just contact us. You will have our information on the website. And again, if you want, you can also contact me directly at my address at SupBiotech. As for today's agenda, so we will be interested in biotechnological valorization of natural products and waste. As I mentioned, we will have 2 seminars, two presentation today. It's 25 minutes presentation that will be followed by 5 to 10 minutes of question. Again, you can ask your question on the chat or after the talk, and we will close with some discussion conclusion on the seminar. So today we have the chance to have 2 speakers. We have Sara Barbosa, who is assistant professor at Coimbra Agricultural Superior School at the Polytechnical Institute of Coimbra in Portugal. She is a specialist in biotechnology and dedicated to agriculture and environment, and she's also project manager for Izeki Food Association. Welcome, Sara. We also have Agnès Saint-Pol, who is professor at SupBiotech. SupBiotech is the Institute of Biotechnology of Paris in France, and she's head of the lab LRPIA, which is an agroecology lab. And her research is dedicated to the valorization of plant waste. So I would like really to thank deeply our speaker. This is our first webinar. It's not easy to start things, and I would like to thank Sara and Agnès for contributing to this first seminar. So now I will leave the stage to Sara. I will stop sharing my presentation and just ask Sara if she can share her PowerPoint.
So good morning, everyone. Good morning to all participants. I would like to thank you to Dr. Patrick and Dr. Martin Henriques for the invitation for this first webinar. So to start, okay. Let me check. Okay. To start, just a little about me. So I have a master in biotechnology. I'm collaborating with CERNAS since 2028. CERNAS is a research center of natural resources, environment, and society. I am an invited assistant at the Coimbra Agricultural School since 2021, and a project manager at the Zeki Food Association since 2023. So just to have a brief introduction of this topic. So let's start with a problem we are familiar with, food and agricultural waste. Globally, we generate over 1.3 billion tons of food waste each year. Meanwhile, farmers are dealing with poor soil health, expensive chemical inputs, and climate change. So, can we use food and agricultural byproducts not as waste, but as a valuable tools to manage in a more sustainable way? So tackling these issues requires global cooperation and innovative solutions. And that's where the global goals for sustainable development come in. They are also known as the Sustainable Development Goals, and they were adopted by all United Nations member states in 2015 as a universal call to action to end poverty, protect the planet, and ensure peace and prosperity for all by 2030. There are 17 interconnected goals that you can see in this picture. And they addressing critical areas such as climate action, responsible production, zero hunger, clean water, and more. And why does this matter? So we rely heavily on synthetic fertilizers and pesticides, but they degrade soil, pollute water, disrupting ecosystems. So reusing agri-waste directly supports multiple and sustainable development goals. So this research aligns with the United Nations Sustainable Development Goals, particularly Goal 2, Zero Hunger, Goal 12, responsible consumption and production, and Goal 13, climate action. So, in crop production, the use of synthetic chemical herbicides is practically mandatory, despite concerns about public health and the environment, as well as the need to meet sustainable goals. So therefore, it is important to find eco-sustainable alternatives to synthetic herbicides. On the other hand, organic farming faces a major challenge: how to control it. So it's here that sustainability meets the circular economy. So is it possible to use agri-food waste to control it? And that's where our case study begins. So the aim was to assess the bioherbicidal potential of 3 agri-food waste: coffee grounds, corn cob, and soybean pod, when incorporated into soil, and their effect on weed suppression in a spring-summer horticultural system with lettuce and radish. So the methodology, we start by collecting 3 composite soil samples at the campus of ESAC, that were where our experiment take place, and we used 4 treatments: control plus the 3 wastes that are coffee grounds, corn cob, and fava bean pod. We did aleatory distribution of the agri-food waste with 4 replicates each in its plots, and incorporated the agri-food wastes 15 to 30 centimeters underground. And we did the plantation of lettuce and radish. After 4 weeks, we collected samples of above ground with biomass in each treatment and plot, and collected samples of soil in each treatment and plot too. After, again, 4 weeks, so at week 8, we collected again samples of above ground with biomass in each treatment and plots and soil, and the crop harvest was carried out manually. Along this time, we did NDVI measurements. So this stands for Normalized Difference Vegetation Index, and it's a widely used remote sensing measurement that helps assess the health and density of vegetation. So here you can see the field scheme, and we have 16 plots in total, 4 replicates each of control. The control were the plots without any waste served as control. And we had coffee ground, corn cob, and fava bean pod. And here you can see the example of each plot. So with the respective agri-food, and then they did that green lines that where we planting lettuce, and the purple where we planting radish. To collect samples and to do the measurements, we used for soil samples a soil probe to collect the samples at all plots 30 centimeters underground. The NDVI measurement was to assess plant vigor and monitor their development. And for with biomass, we used metal squares that you can see at the picture. And then we cut them like you see in this picture on the left. And in each plot, we took 4 random samples, 25 to 25 centimeters, from the central area of the plot, avoiding the edges. And in the laboratory, the wedges were separated into 3 groups. Then they were placed in paper envelopes, drying, and weighted using a scale to determine the biomass of each wheat group, and the total biomass was then calculated. So here you can see the lettuce and the radish, and we separated them in aerial part and root part. So 8 weeks after plantation, crops were harvested, showing suitable development for commercialization. The aerial and root parts were washed and separated, and they were dried and weighted too, to obtain the aerial root and total biomass, and the ratios were calculated based on the edible portion of each plant. With the determination of chemical properties of crops, lettuce and radish, and agri-food wastes that we incorporated into the soil. So, corn cob, faba bean pod wastes, as well as lettuce leaves and radish roots were ground using a mill specifically designed for plant material, equipped with a 0.5 millimeter mesh sleeve. The coffee grounds did not require grinding as they already had the appropriated particle size. Both of the wastes and the plant material were subjected to laboratory analysis to assess their nutrient composition in macronutrients and micronutrients. We did determination of soil chemical properties too. So as I said, we did soil sampling, we did the nitrogen determination, pH determination, determination of organic matter, and determination of extractable phosphorus and potassium. So for the statistical analysis, the measurements of NDVI with biomass and soil analysis, we did a general linear models with analysis factor of interaction, treatment, and time. For crop biomass, crop and waste analysis, we did a general linear models too, with factor analysis, just treatment, and to verify and compare the post-hoc mean values of variables under study were tested using the Taki test, and the significant level was set at p less or equal to 0.05 for all analysis. So, passing to the results. Here you can see the NDVI measurements, and what we could observe was the incorporation of coffee grounds into the soil decreased the NDVI values when compared to the control values, particularly in weeks 3 and 4. In the other weeks, none of the treatments differed from the control, and although the growth of both weeds and crops contributed to the NDVI values, the readings recorded for coffee grounds and corn cob in the 3rd and 4th weeks seemed to indicate reduced plant development, particularly that of weeds. The results about the wheat biomass. So the incorporation of coffee grounds into the soil decreased the biomass of monocotyledons, weeds, and the total biomass of weeds that you can see. So the monocotyledon weeds is this graphic, and total weeds, so the lower graphics. About crop development, we saw that the incorporation of coffee grounds into the soil promoted development of the crops. So you can see that all graphics where we used coffee ground have bigger results, higher results. And about the chemical properties. On the agri-food waste, the coffee grounds waste showed intermediate values with the highest content of nitrogen among the macronutrients and copper and manganese among the micronutrients. In general, magnesium was the most abundant nutrient in the corn cob waste, which was the poorest in terms of the other nutrients. And the fava bean pod was the waste that showed the highest amounts of macro and micronutrients, except for magnesium, nitrogen, copper, and manganese. About the crops, in general, the lettuce crop showed higher values for the analyzed nutrients when compared to the radish crop. In the radish crop, the plants showed a higher phosphorus content on the plot where fava bean pods were incorporated. In the lettuce crop, the plants that grew in the plots where corn cobs were incorporated showed a significantly higher iron content than the plant controls, and a lower content of this nutrient was observed in the radish. Still in the radish crop, the plants had a higher zinc content in the plots with corn cob incorporation. In radishes, copper and iron content were lower, while zinc levels were higher. As for manganese, radish grown in plots with fava bean pod presented higher levels than those reported in this study. About the soil chemical properties, no statistically significant differences were observed in the chemical composition of the soil samples collected from the control and from those collected in the wastes. However, we saw that time influenced the organic matter and potassium content, which are significantly higher at the 4-week mark. When comparing the analysis data with the initial values. There is, before the experiment was set up, it was found that the organic matter content, pH, phosphorus, and nitrogen were slightly higher before the start of the experiment, while potassium showed very similar. So, to conclude this theme, coffee grounds were the waste that showed the best results across all parameters and analysis carried out. The agri-food waste coffee grounds can be considered a potential alternative to synthetic chemical herbicides. The increased development of the crops may indicate an additional fertilizing effect of coffee grounds. And the fava bean pod seems to have a fertilizing effect as it stimulates the biomass of dicotyledonous wheats, and a positive trend was observed in the biomass of radishes. So this study met the objective of the work by highlighting the potential that agri-food wastes, particularly coffee grounds, can have in organic farming systems, which lack effective solutions for weed control. In addition to helping with management, this solution contributes to promoting the circular economy by giving a new use to agri-food wastes. So, to wrap up, waste isn't waste, it's a resource, or it can be a resource. Agri-food byproducts have real potential to improve crop management, to reduce costs, and protect our environment. But to fully realize this potential, we need partnerships between science, farmers, industry, and policy. and together we can close the loop and build a more sustainable agricultural future. So, thank you for your attention. I would like to take this opportunity to thank to Dr. Cristina Galhano and Dr. Paula Lorenzo for their guidance and collaboration on this work, and to Coimbra Agricultural School for their willingness to carry out the experiment on its campus. And feel free to connect with me afterwards if you like to discuss more about this work. Thank you.
Thank you very much, Sarah, for this very interesting talk. I like your sentence: waste is not waste; it's a resource. So we start opening the eyes on this fact, and it's very important. It is important for environment, as you show. It's important for economy, and it's more and more important for geopolitical reasons, because we would like to be more independent, let's say. So it's really a very important subject.
Thank you.
I don't see any question, but maybe I really want to ask you one question. So you mentioned that you didn't see any difference in composition of soil, but you saw an increase of organic matter, right? When you amended. And I was wondering whether the effect you see on fertilization, on reduction of weeds, could be related to the microbes in the soil. Is it something that you check? Do you see any variation, changes in soil microbiome, and could this be correlated with the effect that you see?
We didn't check the microbiology of the soil, so I can't give you a response or an answer about that.
Mm-hmm. Because ultimately, what is causing the effect that you see? How do you explain the reduction of weeds overall in your system?
Well, the coffee ground has a reduction of the weeds, and the fava bean pod has the effect of a fertilizer, probably because of the compounds of each agri-waste influenced the weeds.
Okay. So it's the natural compounds that are within the waste that have this effect.
Exactly, we believe so.
Okay. Did you had chance to identify some of these compounds, and you already have some clues on which compounds are more active, or?
No, we just did the chemical properties, so in macronutrients and micronutrients. So we didn't do the flavonoids or others that could effectively have here a improvement of these properties.
Okay, interesting. So feel free to ask question, whether in the chat or you can ask directly. No, if I don't see any question for now, we will have some more minutes after the talks. But so then I thank you again, Sarah, for this presentation.
Thank you.
I will give the stage now to Agnès for her presentation.
So thank you very much for Patrick for having given me the opportunity to present the work we are doing in the lab. So at SupBiotech, we are working on the valorization of leak bioproducts into an antifungal biopesticides. So first of all, I would like to start with a general context, especially if some students are listening and if they are not really familiar with the topic. So phytopharmaceutical products or phytosanitary products or pesticides are preparation containing one or more active substances for protect plants against any harmful organisms. They mostly encompass herbicides, insecticides, fungicides, and nematicides. Okay, so you probably know that they are extensively used in conventional agriculture since the 50s, particularly in America and Asia, to a less extent in Europe and Oceania. In France, for example, we buy each year, or agriculture use each year around 45 to 50 million kilograms of phytopharmaceutical product each year. Such use has had a very important impact for food safety, and it is estimated that each year the usage of such product limits the 70% loss of vegetables, fruit, sorry, first 50% loss of vegetables and 30% loss of cereal. Amongst these pesticides, fungicides represent almost half of the pesticides used in agriculture. This can be correlated with the prevalence of fungal disease in plants. If now we estimate the plant loss due to disease, this represents between 10 to 50% of production loss, which is not so much. But if now we look to this loss, more than 70% is due to pathogenic fungi, which explains why fungicides are so used in agriculture. In terms of volume, it is quite important because each year it is estimated that around 125 million of the 4 major crop production are destroyed. This is rice, wheat, potato, and maize. Unfortunately, you also probably know that such practices have a cost, and notably for diversity. Just to illustrate this, I would like to underline a study which has been performed in France after that French government has commissioned these 2 public research institutes, INRAE and IFREMER, where there about 50 researchers really studied more than 44,000 scientific papers to evaluate the impact of the phytopharmaceutical product on biodiversity and ecosystem service. The conclusion was that effectively the PPP have a very negative impact on biodiversity. They are present everywhere on the surface of the planet, and there are still a lot of studies lacking of such effect on biodiversity. So finally, to conclude on this general context, we can see that on one hand, crop treatments guarantee food safety, but on the other hand, if we want to maintain biodiversity, there is a urge to change agricultural practice. One solution could rely on using or changing or modifying these phytopharmaceutical products. In this context, biocontrol could be a promising solution. So when we present biocontrol, we refer to 4 different categories of products. Microorganisms as predatory insects, microorganisms as fungi or bacteria, chemical mediators, or natural substances. Our lab in SupBiotech focuses its research activities on natural substances, and we are interested in particularly a category of natural substances, the byproducts, the plant byproducts, and more especially on vegetable byproducts, which are generally not unexplored or not exploited. So we wanted first to evaluate if such byproducts could be an alternative to phytopharmaceutical products. For that, we screen the antifungal activity present in different types of vegetable byproducts. Few of them were quite efficient and prevent the growth of our fungal model. And we finally pay attention to leak byproducts. The reason why we paid attention to such products was, I would say, that for two main reasons. The first one is that Europe is a very important leek producer, as leek is almost daily consumed in our food. Belgium and France are the main European producer of such food, but Poland, Spain, and Germany and Netherlands produce also a high quantity of leek. The second reason why we pay attention to leek byproduct is that this production in terms of volume is quite high. When leek are harvested, they are treated by peeling and cut to remove byproduct, which at least is a mix of the outer white part of the leek and the extremity of the green part. Next, today, at least in France, these byproducts are treated as waste after having been collected in such tank. In terms of percent of leek production, such products represent about 20 to 15 to 20% of leek production. As we have seen previously on this antifungal activity, leek is also known as a good candidate because it contains some antifungal molecules or antimicrobial molecules as organosulfur compounds, saponins, and for example, boric acid. And we have recently isolated a new candidate. So in our study, we wanted first to really compare the activity of leek byproducts on different fungal phytopathogens. So we screen our leek extract on different pathogens as Alternaria alternata, Fusarium, Penicillium, and Cladosporium, and even Botrytis cinerea. What we've seen is that whatever the fungus targeted, our leek extract had activity and decreased the growth of the fungi. In the next experiment, we have focused attention or used Alternaria alternata as a model because in vitro, it's possible to mimic the infection, the plant infection of this fungi. First of all, we could evaluate the binding or the attachment of Alternaria alternata onto surface, which is correspond to the first stage of plant infection by such fungus. Observed is that leek byproducts extract inhibits these attachments. So we can see that on the control, after 60 minutes binding conditions, Alternaria attached to the plastic surface, and in presence of leek extract, this attachment is prevented. At low concentrations, there is a delay of attachment, but at higher concentration, even after 5 hours, it was not possible to see any spore attachment. We next wanted to evaluate if leek byproducts extract could inhibit germination of spores. So in this experiment, we first induce spores attachment on the surface and after treat the spores by our leek extract. After this attachment stage, some spores had already germinated, which gives a bit of background. So what we can also we had observed is that at low extract concentration, we had a delay in spore germination in comparison with the control, but at higher concentration, the germination was inhibited, if at least up to 2 hours and a half leak treatment. The next stage is hyphae elongation, and to evaluate the impact of leak extract on this elongation, we treated the alternaria after different growth stage, as either germ tube, early hyphae, or late hyphae. We then observed that whatever the stage of the fungal growth, the leak extract was active at high concentration, but at lower concentration, the leak extract was more active on spores than on germ tubes, early hyphae or late hyphae. So finally, what all these first experiments indicated is that the leak extract prepared from leak byproduct was active against all the different stages of fungal growth, but the activity decreased after late development of the fungi. We wanted next to investigate the effect of the leak extract on this hyphae. And for that, we incubate hyphae with leak extract and let the fungi grow. And we observed, have to wait like a few seconds, after like 2 to 3 minutes hyphae treatment, we observed a very important explosion at the apex of the hyphae, showing that the removal of cytoplasmic content and explaining the effect of leak extract. Okay, so we wanted next to increase the investigation of the mode of action of the leak extract and evaluate this activity on alternaria spores. So to evaluate the death, but also the loss of permeability of the spores, we performed microscopic experiments using iodide propidium, which is incorporated in spores only after membrane permeabilization. So you can see in these pictures that in comparison with the control, the leak extract treatment promote a strong iodide integration. This phenomenon is quite rapid because it is possible at high concentration of extract to quantify or to see that around 50% of a spore population is already permeabilized after 3 minutes leak treatment, and more than around 80% of this population is permeabilized after 2 hours and a half treatment. This loss of membrane integrity could then be correlated with a rapid oxidative stress. To evaluate such oxidative stress, we used a probe, the DCFHDA, which become fluorescent after exposure to oxidant. So same things, we can observe that in comparison with the control, the presence of leak extracts induce the apparition of fluorescence. Again, this phenomenon is quite rapid. As you can see here, after 30-minute leak treatment, half of the population is undergo an oxidative stress at high concentration, and we can observe a dose-dependent effect. After 2 hours and a half, what is quite interesting is that the population undergoing oxidative stress is decreasing at high concentration, still increasing at lower concentration. So it might be due to a kind of wave of oxidative stress, which leads to loss of membrane integrity. So finally, to really correlate both oxidative stress and rapid loss of membrane integrity, we exposed the spores to antioxidant and acetylcysteine and to evaluate the effect of inhibition of oxidative stress. So in these slides, you can observe that the treatment with extracts combined with N-acetylcysteine lead to an inhibition of the loss of membrane integrity. We quantify this phenomenon by measuring the biomass of Alternaria alternata after 24-hour growth. And even if N-acetylcysteine delays the growth of the fungus, you can observe that at high concentration of leak, it's allowed to have growth comparable to the control, showing that N-acetylcysteine prevents the leak extract action or antifungal activity. Okay. Finally, we wanted to evaluate if such leak byproduct extract could have a potential in plant protection. So for that, we infected seed with both two fungi, either Alternaria alternata, our model, or Botrytis cinerea. And after 7 days of culture, these fungi really delays, or at least damage or even kill the seeds, the plant, whereas the treatment with our leak extract prevent lead infections and leak damage. We also did combine this approach by another one, which consists in combining the leak extract with other phytopharmaceutical substances. The idea behind is to increase the activity of the leak extract, but also to be able to decrease the quantity of phytopharmaceutical products. For that, we screened for synergistic effects of leak extract combined with such products. So to evaluate the synergetic effect, we performed some isobologram in which we plot the two doses of substance, actives or drugs, and measure the combining action of both. If they act synergistically, we can observe this kind of graph. So some different drugs have been tested or different combination, and some of them really show a synergetic effect, particularly the combination of leak extract with surfactants. So to conclude, on the main point of these studies, we have seen that the leak extract obtained from leak byproduct inhibits early stages of fungal plant infection, from alternata attachment to early elongation. This inhibition is associated with a rapid oxidative stress, which is comparable to typical antifungal drugs, which generally promote oxidative stress. And we have also observed that as a leak extract obtained from leak byproduct protects broccoli seed from at least two fungi, Alternaria alternata and Botrytis cinerea. And finally, it might be possible to potentialize this inhibitory effect by combination with surfactants. So we have seen in the general introduction and that phytopharmaceutical products have negative impact on biodiversity. So we started experiment in the lab to evaluate the impact of leak byproduct on soil. Two approaches have been used for this approach, a culturomic approach and a metagenomic approach. What we can see in comparison with control is that leak extract modulate the bacteria the bacteria population in soil and seems to have a very strong impact on fungal population in comparison with other phytopharmaceutical products. In terms of abundance and diversity, the leak extract and for bacteria does not have really impact on phylum, but surprisingly, the leak extract increase or enhance the bacterial biodiversity in soil with some population appearing after soil treatment by the leak extract. So all this study is still ongoing, but these results suggest that these products could modulate the soil microbiome. Okay, so just before questions, I would like to thank the different funders. So Europe, the SupBiotech and SupBiotech. And finally, I would like to thank all the members of the team for contributing to this study. So thank you for your attention.
Thank you, Agnès. Thank you very much for this presentation. The stage is open for question now. I don't see any question in the chat. We can start a general discussion together with Sara as well. So I would like to invite you, Sara, also to connect. This subject of reusing waste is very important, as we mentioned, and you clearly illustrate here. And one question that I have is that there might be some resources available, but they are limited, right? So my question is, do you think that this valorization that you are speaking about and illustrating, can we valorize at the local scale, at the regional scale, at the national scale, at the international scale, or what would be the best scale to make this valorization?
What do you mean exactly, please?
I mean, for example, you mentioned that there is 15 to 20% of leak byproduct.
Mm-hmm.
So it's a lot, but at the same time, it's just a finite amount of leak byproduct. Could we imagine to use this leak byproduct at the international scale, or there is not enough, and we have to think about using this technology at a local scale or national scale?
Local scale.
Yeah.
Local or national.
Yeah, I think, yeah, if we really want to really for in case of perspective of sustainable development, I think it might be much more interesting to use them as a local scale, but the approach can be reproduced in each country which produce such products.
Yeah. In our case, in our experiment, we used the agri-food waste of coffee ground, and this is used every day in all cafes. So probably national, but about corn cob and fava bean pod, it's agri-food that is more reduced, so probably more local.
Also, just to give a bit more details about the work we are doing in the lab, we are currently have funding to work with the Normandy. So Normandy is a French area which is specialized in the production of vegetable, and we are performing a study to evaluate the protection of the leek extract on local plant disease, and particularly to evaluate the impact of the leek treatment on the protection of lettuce, sorry, and carrots. So the idea would be to reuse local waste to protect for our project local vegetable.
Thank you for your feedback. We have now some question appearing in the chat, so I will deliver this question. So first question concerning, so the question is, have you tested the effect of leek extract on some crop species? You just mentioned that you are doing a study on carrot and lettuce.
Yeah, exactly. So we have started the tests at the autumn, so we don't have the result yet. But yeah, the idea is to try to evaluate if the leek extract can protect les tooth, sorry for pronunciation, from mildew, and carrot from alternaria and pythium disease.
Yeah, thank you. There is another question from Piotr Garonsky, who is wondering what metagenomic experiments were used to analyze bacteria in soil.
Okay, so actually, Patrick, you will have more the answer than I.
Yeah, so what we did is 16S RNA sequencing that we perform on the different treatment that Agnès was showing in the same condition for everything. And we just, for the moment, measure the relative amount of each type of genus and phylum, and we are also getting the results for the fungi now. Another question is what are the implications of leek byproduct extract application on soil, microbiota dynamic, soil health parameter, particularly regarding non-target microbial population and long-term ecological balance? Yeah, so that's very interesting question. You want to answer, Agnès?
No, you can answer. I think it's more your field.
Okay. Yeah, that's a very this is a very interesting question for us. As Agnès mentioned, we are trying to limit the damage to the environment, so we don't want to make more damage using our product, and it's not because it's natural that it's without danger. So we are measuring this on bacteria and on fungi. As Agnès showed you, it seems that the fungal population is deeply impacted by our treatment. The impacts are less for bacteria, and what we want to check is the kinetic, as you mentioned here, the kinetic effect. Do we have a resilience of the population, or is it long-term effect that we are causing here? We don't have the answer from now, but this is something that we are taking seriously in the lab, and we are making the experiment to get the answer.
So just I can add that for this experiment, I think Patrick did a treatment by leek extract and recover the population soon after the treatment. But what we know is that the leek extract activity is not it's not really stable, and it degrades quite rapidly. So I suppose that really we have to make the same experiment, but after middle or long-term effect, because it's likely that there is no accumulation of active molecules in soils because they are not really stable.
Yeah, from now, what we can say is that we are not losing bacteria. We are bringing more genus of bacteria. Whether this is good or bad for the soil, we don't know yet. One of the genus that we identify has been correlated with some biostimulation effect in some publication. So in that way, it could be a good thing. But yeah, we really don't know. We don't have for the moment strong answer on this. Well, I can ask a question. So we can see that things there are things that are possible, application that are very interesting that we can do application from waste. But what are the breaks and the challenge that we have to remove to really make a real application out from your research, for example?
I think one of the challenges is that it is quite well described that biocontrol agents for the project we are performing are generally less efficient than conventional phytopharmaceutical products. So I think that they can be very useful, but if their use is combined with other approach or other modification of agricultural practice. So one of perhaps of the challenge is that farmer really modify their global approach of agriculture if we want to really introduce such product in agriculture. I think
the point of view of farmer is important.
Yeah.
What's the feeling of farmer according to your study, Sarah? How are they interested? Do they want to implement such kind of approach?
Regarding your first question, I would say that a big challenge is to catch some farmers that could be available to do this experiment in their fields, and then say to us if they have the same results as us. So yeah, the agri-food waste can be a challenge on that, I think.
Mm-hmm. Yeah, that's very important. It add a new dimension. In addition to the technological and technical dimension, we have to have also this social dimension where we get involved the farmers in the project. I think it's very important to get them involved as soon as possible in the project.
I have a question for Sarah. So you did your analysis on the soil on after having added like different waste, but I wanted to know if you had studied the composition of the waste at the beginning, like the coffee ground.
Yes, so we started doing collecting samples of the soil without any waste and with wastes, and we analyzed the agri-foods by ourselves and incorporated in the soil.
Okay, and the coffee ground, where the origin of the coffee ground was?
It was the cafe of our school.
Okay, okay, so you collected the
yeah, okay. And home too.
Okay, so it's a mix of perhaps different origin of coffee.
Yes, we did a mix, so we don't know the composition, the exactly composition of the coffee ground.
Okay. Thank you.
You're welcome.
Yeah, this makes me think that maybe one other challenge is to collect the waste. This might be also kind of challenge.
Yes, in our case, we needed to mill to ground the coffee grounds and the farming pods, so that needs to be in a bigger scale. That needs to be industrial, maybe. So yeah, it's another challenge.
Yeah, that's yeah, we see that there is potential, but it means that we also have to reorganize basically the way we are working in our society. So we have to look much much bigger than in our lab. So we have to think about society. Yeah,
yes, effectively, we need to scale up some steps.
Yeah, sure. Because the potential is here, so we have to find a way to really use it. Question to Sara. So somebody is wondering which method did you use to do the nitrogen determination in soil?
Yeah, it was Gjedel method.
Okay, there is another question. Could the fibrous waste from leek harvesting, so this is for you, Agnès, like the outer leaves and the roots, could they be processed into bioactive extract for the use of bioplastics or biodegradable packaging?
I think it might be a good idea because when we prepare leek extract, we still have high volume of back product, which are all the insoluble parts. So it could be effectively a good idea to extract the soluble active molecules and develop another approach to recycle this solid part, which is really enriched in fiber. So yeah, it could be possible, I think. We thought about also use them to promote biogas or to, but it making some material also might be very interesting.
Yeah. If I may add, at the genesis of this project, the idea of Agnès was to incorporate leek extract into plastic to protect fruits and crops when they are into plastics. But we found that there is no volatile molecule, antifungal volatile molecules in leek byproduct, so it doesn't work. There is another question from Miguel. What about brassica waste used as phytochemical? The relative high content in sulfur could be effective against fungal soil-borne disease.
Yeah, that's true. I don't know if you remember, but when we did the screening, we tested cauliflower and turnip peel, and clearly turnip could also be a very interesting candidate. Yes, likely due to its organosulfuric compound. But finally, as I mentioned, we focused our first study on leek because of the volume of production and the availability of byproducts. But sure, brassicacea could also be very interesting.
Yeah, and in terms of waste amount, that would be also a good target.
Maybe it could be another project.
Yeah,
for the next years.
we are recruiting who wants to start. Yeah, that could be also. Or we can also think about making a mix of the available waste in a region, for example, to make some kind of mix that is more active. This is something that can be think about.
Yeah. But the beginning of really this project was to focus on byproducts which were not explored or exploited, and at least in France, this is the case for vegetable byproducts, because I thought that it might be due to the, in comparison with cereals, for example, the low volume production annually, because French company really developed some process of valorization of industry like cereal industry, beer industry, but not these sectors as vegetable production.
So one big challenge that we are facing, and probably you also, Sara, is to identify the active compounds. This is
yeah, exactly. It could be done, yes, and the soil microbes.
Yeah, as mentioned by Miguel, there are also, of course, the usual suspect like sulfur compound, but there is much more to be discovered, and we have found new substances that we cannot disclose now. But yeah, there is more than what we know now. Yeah. So a lot of work for students in particular. If you want to engage into this field, there is a lot to do.
Yeah, and in terms of organosulfur compounds, we have developed in the lab a test to evaluate the contribution of volatile compounds in an extract, and so we have measured the. We have evaluated the contribution of volatile compound in leek and compared the activity of volatile compound in garlic, another Alliaceae, and we were surprised to see that if there is antifungal activity coming from a volatile compound in garlic, apparently there is no volatile activity associated with leek, which is not a proof, but which suggests that organosulfur compounds, if they play a role in the global activity, it's a minor role.
They are from the same family, but apparently the active compounds are not the same.
Yeah, exactly.
If it's fine for everyone, so then I just want to thank again Sara and Agnès for their presentation. Thank you very much.
Thank you for the invitation.
It was very interesting. As I mentioned, the next webinar might be on the same topic, different subject, but the same topic. So I kindly also invite you to the next seminar. And I just want to share one more slide. So I would like to invite you to answer this survey about this webinar. This will help us to improve our webinar. This is the first from the series, so we greatly appreciate your feedback. And again, I thank all the speakers today. I thank you also as an audience to support our webinar, and I thank Agathe, who helped me a lot in putting together this webinar. And with this, I wish you a nice day. And again, if you need to contact us, go on the UNIgreen website or contact me directly on my email, and we will be really happy to answer your question. Thank you very much, and bye to everyone.